Perfect. Okay, thank you. Thanks to Trey and Nevin for putting on the symposium and for the invitation to speak. I realize I'm the 5th speaker in the longest session of the day, so do my best to move it along. So I'm going to tell you about networks of non-coding mutations in cancer, and to set the stage, I think, as we've already heard, the coding landscape in cancer has been pretty well analyzed, most recently through the TCGA, and in the last sort of analysis of the TCGA PanCan Atlas project, which, among other things, sort of produced this list of 299 driver genes by sort of combining various prediction methods to find recurrently mutated genes. Unfortunately, that's not quite perhaps the list of all mutations that drive cancer, as we all know, and there are a number of issues. The first, of course, is one we've sort of already heard about in the first talk of this session, which is this phenomenon of the long tail, where if we rank genes according to their sort of recurrence, or here their sort of statistical significance of recurrence, then we draw the line of statistical significance somewhere, and we sort of miss genes that are too rarely mutated in our cohort to be statistically significant on their own. So this long tail phenomenon means we're still probably not characterizing all the cancer-causing genes and cancer-causing mutations by recurrence. And then, of course, there's the issue of, what about the other 98% of the genome, the non-coding genome? And that's what I mostly talk about today, and talk about some results from another sort of consortium project that I was involved in recently, and this is the pan-cancer analysis of whole genomes. So this is taking approximately 2,800 whole cancer genomes across a couple dozen cancer types, and really focusing in on what are the mutations in the non-coding regions of the genome that might be driving cancer. So, from sort of a first view of this data, if you run many of these same driver prediction methods based on recurrence, you see that when you look at the non-coding picture in the same way, the long tail's gotten much, much longer. And, in the coding space on these same genomes, we can sort of recapitulate many of the known cancer-causing genes. Here, there's the cancer genome census. There's still some that we're kind of missing. But in the non-coding space, we have, TERT promoter mutations and hardly anything else that's sort of significant. So, this is a real problem, and to some extent, a real challenge of this project, which was, sort of hoping to really find the additional non-coding mutations that were driving cancer. But of course, for this audience, we know that perhaps looking at one gene at a time is not the best way to analyze this data, and we should perhaps look in combination, and we should look at networks. And this is, pretty well worked out in the coding space. And, another picture from a TCGA project, and I think Trey had head and neck cancer. Here is colorectal cancer. But again, the idea of mutations in the coding space aggregating on networks. So can we do the same type of thing in the non-coding space? So, quickly, just as a way of reminder, I wanted to sort of say that, this is not actually such an easy computational problem to take mutation data measured at the gene or protein level, or in our case, sort of the non-coding elements that might drive gene expression, and a big network, and try to sort of put those things together in a way that's reliable and robust. And there's many challenges, and in particular, we've heard a few already in the session, and these networks are very diverse, very heterogeneous. They have these, hubs of high degree, and this sort of small world phenomenon where everything's connected to everything through a few hops. So you really have to do this carefully and robustly. And there have been many such methods that have attempted to do this. We heard about NetSig, which is here. Trey's group, of course, has done a lot of nice work in this area, and our sort of contribution to this is an algorithm that called HotNet, which sort of takes mutation data, uses that as sort of an initial distribution of heat on the nodes of the network, and then diffuses that heat for some length of time. And so what that does is sort of naturally smooth these scores over the nodes of the network, and allows us to sort of pick out these hot spots on the network, which are driven both by the original scores on the genes, but also the connectivity between these genes in a very natural way. And so, we were intending, and we started to apply this in the non-coding space, and, we always had a few kind of nagging issues in these types of methods that we really wanted to revisit as we were doing this new analysis. And, two of the, I think, really nagging issues that face this work, this type of work computationally, is that if you actually look at the data, this is again, the TCGA PanCAN Atlas, so the coding mutations. If you look at that data just in terms of, the highest frequency mutations or the most significant in terms of a statistical correction, significantly mutated genes, there's actually this pretty strong correlation between degree in the network and frequency or significance of mutation. And so what this means is if you go run a network method and then try to run some statistical test that tries to randomize, and you don't account for this correlation, your results are going to look hugely significant, much more significant than they should be. And so it's really important to be aware of these types of hidden confounders when we're doing this analysis. And then the other sort of issue that we'd always sort of faced, and a number of these sort of methods face, is that, there's always some sort of hidden parameter that needs to be set to decide where exactly to kind of draw the line and where to kind of break up the network into these constitutive subnetworks. And often this is something that's really hidden in the supplement of the publication, and, it's set to some value that might be, set in a reasonable way or an unreasonable way, and that value might change with your data, and it's not really so clear how that should be done. And it's actually quite an important issue because, depending on where we sort of choose to break apart this network, we might be, getting predictions that are at the really fine scale of protein complexes, or maybe sort of a larger scale of proteins, or, really big connected networks at the level of sort of processes. And it's not often so clear where exactly to draw this line. So we sort of went back to into the methods development part of the group, and sort of develop a new method that, continuing our sort of naming convention, we call sort of hierarchical hotnet. And this is really to address these two major issues. And the first issue of this statistical significance, I won't really describe too much in detail. I'll just sort of say that we have a permutation test that's really aware and cognizant of this correlation, and really preserves this correlation between degree and score as it does randomizations and as it does the statistical test. But the second part of finding the appropriate biological scale, we actually realize that we don't have to make a hard decision on where to draw the scale. And in fact, what we can do is we can produce, by changing sort of the parameter of the network, a hierarchy of subnetworks as we vary the parameter, and really develop a statistical test to sort of assess that whole hierarchy and sort of find the range of subnetworks across these different scales. So, without going into details in the first step, just to sort of illustrate the point here, depending on, once I combine this information between the gene scores and the network into sort of a hierarchy, and you can think of this as just a version of hierarchical clustering, then depending on where I decide to make the cut, I'm going to get subnetworks of different sizes. So I can just, choose a parameter that cuts down here, or I could be more permissive, cut up here, and I'll get sort of a continuum of subnetworks from individual nodes all the way through to the whole network. And what we can do is take that idea and kind of wrap that up in a statistical test that will actually, on permuted data, also build these hierarchies and then actually compare the two different hierarchies. So we don't have to choose a parameter. Instead, what we can do is we can look at the hierarchies that we get as we vary this, where we cut across the continuum. On randomized data, we get sort of subnetwork sizes, that are down here. And on our observed data, we get this, these much larger subnetworks. And then we can look at, where is the range of parameters where we see a difference, if at all. And we can also develop a statistical test based on a maximum statistic to decide whether or not this deviation we're seeing is significant or not. So this gives us a way of really just looking at the data almost directly and seeing whether there's signal. And so that on this now non-coding data from ICGC, if we look at, from the PCOG project, the coding and the non-coding data. And the coding, what we sort of see is as we, sort of vary this parameter from, individual nodes all the way up till, a massive large subnetwork, so the whole network. On randomized data, these blue lines, the observed data in red with scores, and you can see this significant deviation. So this shows you not only that there is signal, but actually shows you where the signal is in terms of the scales of the network. In the non-coding space, we also sort of see a signal, but you can see actually that the signal is quite subtle. It's almost hard to see the deviation, but it is there, and we can test this robustly. So We used hierarchical hotnet along with several other, 7 in total, pathway and network methods to look at the PCOG data, so looking at both driver mutations and non-coding mutations in promoters, UTR regions, and enhancers, sort of put all this together. We ran 7 different methods. We built a consensus. In the end, we came up with 87 what we call pathway-implicated driver genes according to their coding scores, and 93 according to their non-coding scores. And so looking in this sort of view of kind of the long tail, but here in dark are the, our pathway-implicated driver genes, in light are not. If we look just by driver p-value, we're recapitulating most of, the ones that are significant at this FDR level, but also we're finding stuff in the long tail. So these dark bars are sort of things that are sort of coming out because of their connectivity in the network with other coding driver mutations. And in the non-coding space, where really TERT was the only thing that was significant on its own, we can, of course, almost everything's in this long tail, and we can start to pick out these individual genes. We can compare them to known cancer-causing genes in the cancer gene census. They're in blue, and of course, a bunch of them overlap. That overlap is significant. And even in the non-coding space, we're finding non-coding mutations in cancer gene census genes. But of course, that's really just one way of evaluating the data, and if we just look at what's known already, we won't by definition discover anything new. So we spent a lot of time to try to say, could we in some way, using other data, try to provide some other external validation of these non-coding predictions. And so one of the things we did was look at the associations between mutations in these non-coding elements and changes in expression in the same gene. And if you do a simple test like that, of course, the top thing that comes out is the TERT promoter mutation. Well known that promoter mutations in TERT drive overexpression of TERT, and that's the strongest signal in the data and easy to find. However, there are a few other non-coding mutations of this type. Sort of second on the list are actually promoter mutations in P53. So we find in 6 samples, so it's a very weak but significant signal that promoter mutations in P53 lead to underexpression of P53. And we find a couple of other transcription factors where, again, promoter mutations in only a handful of patients, unfortunately, but robustly lead to underexpression of that same gene. And these are both sort of interesting transcription factors for those individual cancer types. So next, we just tried to sort of put the information together and see what is the relative balance of coding and non-coding mutations and perturbing various biological processes. And here's this sort of the usual kind of hairball diagram, but where each gene is labeled according to its proportion of signal that was from coding mutations in green or non-coding mutations in yellow. And what we can see is that some of the core processes like proliferation and cell cycle, et cetera, are driven primarily by coding. A few obvious outliers, P53 promoter, as I mentioned, TERT promoter, both in the coding and sort of non-coding space. But there were other processes like splicing that seem to mostly be driven by non-coding mutations, although there are, of course, coding mutations that are well known in splice factors, and then some developmental processes that seem to have a mix of coding and non-coding contributions. Looking at this sort of in another way, in kind of a matrix view, here's our almost 200 pathway-implicated driver genes. Here's the different biological processes. So in green are the coding, and yellow are the non-coding. You can see splicing that comes out very strongly as non-coding, core proliferation, chromatin in green, development as sort of mix of the two. And so that's sort of another way of looking at the data. The splicing, of course, was very interesting to us. We spent a lot of time trying to see if we could find downstream targets that were actually driven by these non-coding mutations in splicing. Unfortunately, that was very challenging, not surprising, because actually even in the coding space, coding mutations and splicing factors don't seem to have very specific signals in terms of changing splicing in particular genes. However, there was actually through the PanCan Atlas, a recently published gene expression signature that showed actually, 3 clusters of gene expression in different pathways. You can sort of see them here, driven by coding mutations and splicing factors. And what we're able to do is actually recapitulate those signatures using the non-coding mutations in some of the same, but also different splicing factors. So here's sort of the 3 clusters, and you might be able to see if you, sort of squint, the green dots are coding mutations, the yellow are non non-coding. And so we're grouping some of these non-coding mutations and splicing factors seem to be having similar gene expression signatures as some of the coding mutations that are well known in these same splice factors. So the summary for this PCOG analysis, so the network analysis really helping us amplify a very weak non-coding signal, hierarchical hotnet, sort of filling in some of the issues with some of the previous methods for doing this. Some newly identified genes, but everything here is very rarely mutated, so very difficult to get external validation without going and doing an experiment, but there's some promising sort of targets, and then both coding and non-coding mutations seeming to have kind of differential contributions to perturbing these pathways. I think maybe there's 2 minutes, so in 2 minutes I wanted to just sort of preview some unpublished work, and this is really kind of looking at some of the limits of network analysis. And in this room, I think we're all big believers in network analysis, but as scientists, we should always be skeptical, right, and make sure that we're not just doing things that are wishful thinking. And so as one sort of view of this, I think this sort of paradigm of taking measurements of genes or proteins, and here are the measurements. This might be differential expression. It might be frequencies of somatic mutation. And if we look at this distribution, we might say, is there any outliers here? And you look and you stare and you say, maybe there is something here. It's pretty weak. Right? We can run a statistical test, a t-test, whatever, and try to find these, but we might not find so much. And so, can we use the network to sort of inform this signal? There's a very old idea, right? That's why I showed this in black and white, right? It's a very old idea, and I think one of the first sort of groundbreaking work and sort of showing this idea is work of Trey Adeker, not surprisingly. I think this is when he was a postdoc or fellow, and this idea of what's now been called J-active modules, that you can sort of amplify this signal by seeing that most of the outliers are actually close together, or maybe even connected on the network. So if we look at this more carefully, we'd see that this outlier distribution is sort of connected on the network. But when and how does this work? And of course, if this outlier distribution is very large, or this network is very large, then or the number of outliers is very large, we don't even need the network to do anything, right? If we look at this sort of distribution, you can really by eye pick out the outliers, right? So you don't need the network, right? And of course, if the signal is really tiny, the network's not going to help you at all. The signal's so small that you can't find anything. So where is actually the intermediate where the network actually helps you amplify the signal? I think we should always be sort of looking at this question and asking this question. And we, without going into a lot of details, have been really kind of trying to probe the limits. And I'll just sort of show you this one plot. Here's the sort of separation between the two distributions. When the separation is large, the classic J-active modules and some other recent methods, everything can find the signal. In fact, single gene test, which are in dotted lines, would find the signal. As the distributions become closer together, everything crashes. But, in fact, there's sort of differences in how you model the signal that seem to be making a pretty big difference in how you can pick it out. And the difference between the dotted line and the solid line is where the network helps you. So there's just a little bit of gain here that's sort of apparent from this picture, but it's real, right? So it's, real but tiny. And, we're sort of looking into what exactly does this mean for how we should use networks and what type of networks are the most useful. And one of the findings, of course, is that as the network gets more dense, it doesn't really help you much at all. And so we really need these context-specific networks that sort of have the appropriate level of sparsity that's sort of matched to the data. And I think that's sort of what the various mapping initiatives hopefully are going to be producing, which then coupled with the algorithms will really give us the real gain in power. And so I'll conclude there. Everyone's standing up, so that means I've gone on too long. And just thank the folks that contributed from my group. And then the PCOG was a consortium project co-led by Josh Stewart, myself, with key contributions from Yuri Meiman and many others. So thank you.
Time for one or two questions.
I was going to make a comment about splicing mutations. It turns out that some work that's been done by one of my colleagues, Benoit Palencade, showed that, in fact, our loops are occur in DNA, and if R loops occur, in other words, if the RNA is not removed, this causes major problems with the transcription and, not transcription, but RNA and replication, and you end up getting more genome instability.
Uh-huh.
So and in fact, splicing helps prevent that. And so maybe by screwing up splicing, what you're doing is you're increasing the number of R loops inside the cell, and therefore getting more genome rearrangements, which was one of those arrows on the picture.
That's really cool. I'd love to talk about that further.
It might be interesting to look
Yeah.
at those cancers to see if they're more.
Yeah, that's great, and we have the data to do that, so it would be a great correlation that we haven't looked at.
Hi. 2 questions. One, when you were looking at the TP53 on the non-coding, does it happen, like, when you have a non-coding mutation, do you also tend to have a mutation on the actual gene? And then a second question is kind of unrelated when you were talking about the context-specific work. Another thing with networks is a big challenge is another parameter with thresholding when you have probabilities of interactions or when you have weighted networks.
Yeah.
Can you comment on that at all?
Yeah, so first with the P53, it was only 6 samples that had these promoter mutations. And I don't remember the exact numbers, but a couple of them seem to have other inactivating mutations in P53. So, but not all. So, it was not completely exclusive, but seemed to be explaining P53 inactivation in at least some of the samples. Regarding the networks, yes. The weighted, if you have weights or probabilities on the edges, then that's another way in which this parameter selection becomes important. With this hierarchical method we have, we can actually just encode that same information in the algorithm. It goes in just quite naturally. You could use the weights and the scores, and they just sort of merge together into a sort of unified similarity matrix. And yeah, when you have weights on the edges, it's another way that it creates this issue with how exactly do you decide where to draw the line with the subnetworks.
Thanks.
Thank all the afternoon speakers.